So today in enzymes, we'll be doing inhibitors. Inhibitors basically inhibit the enzyme action. They slow down. So there are two types of inhibitors. One is competitive inhibitor and the other one is non-competitive. Competitive inhibitors bind to active site. As the name tells us, competitive inhibitors, they actually compete with the substrate. They have shape, these inhibitors have shape similar to dash. Can you guess? So bind to dash. Can you fill these gaps? Similar to substrate, mashallah. Similar to substrate, yeah. So they bind to active site. And are in competition. Competitive inhibitors, your chat is here. Active site, very good. Competitive inhibitors are in competition with substrate. So at dash substrate concentration, competitive inhibition will end. And इसमें जो होता है कि Vmax can be achieved. Allow me to explain you after filling the gaps. Similar to substrate and bind to active site. Are in competition with substrate. So at high substance concentration, Vmax can be achieved. Now, what is Vmax? Vmax is when all the enzymes got busy with substrate. So imagine this is an enzyme which will bind to substrate. This is enzyme. So there is a substrate that matches its active site. This is the substrate for this enzyme. But there's an inhibitor which also matches its size, but that is inhibitor, for example. So basically what happens is inhibition occurs when inhibitor binds to substrate. For example, this is enzyme. So rather than substrate, inhibitor could bind here. So this is because its shape is similar. But if you increase the amount of substrate too much, for example, there are too much of substrates. So inhibitor will find it very less a chance to bind to active site if we increase the substrate concentration too much. So at higher concentration of substrate, we can assume there's a possibility that all the active sites are busy with the inhibitor, with the substrate, sorry. When all the enzymes are busy with substrate, this is called Vmax. Vmax is when all the available active sites are busy with substrate. So, when you provide so many substrate molecules, then for inhibitor, it will be very less a chance to bind to substrate. So we said that Vmax can be achieved. Did you get it, beta? सब बात समझ आ रही बेटा ये? Good. So next we do non-competitive. अच्छा इसका ग्राफ भी हम बना सकते हैं. इसका competitive का जो ग्राफ होता है ना वो यूं कि अगर आप substrate concentration बढ़ाएंगे तो देखें this is so when you increase the substrate concentration rate goes up. So if you increase the substrate rate increases, it means the inhibitor was competitive. So it was competitive inhibition. If I draw two graphs, so this inhibitor was competitive because आपने substrate बढ़ाया, rate बढ़ गया. ठीक है? अगर substrate बढ़ाने से rate बढ़े, इसका मतलब competition है, and that is competition is won by the substrate. But there's another inhibitor which is called non-competitive. So this binds at a place other than active site. It is not similar, it does not resemble the shape of active site, shape of substrate. So this binds at a place other than active sites. Binds at some place other than active site. We also call it allosteric site. Allos is different and steric is place, shape. And secondly, there's no competition with substrate because shape is not similar to substrate. So the inhibitor, if I show by graph, if I show by diagram, so imagine this is enzyme. This is its substrate. Which will have to bind to active site. But inhibitor is like, this one is inhibitor. So it binds at a place other than active site. And this distorts the shape of enzyme, so enzyme cannot work well. So no matter how much you increase the substrate amount, unless this inhibitor leaves the enzyme won't work. So some enzymes to which non-competitive inhibitor is bound, they will not be able to work unless you remove the inhibitor, no matter how big is the concentration of substrate. That's why we say that Vmax can't be achieved. मतलब आप इसको यूं समझ लें. If I say, for example, my head, Allah, very late, really. What late हो गया? Imagine this is, for example, my head is all the enzyme. From here above, it's globular sphere. My mouth is the active site. So when substrate has to bind here. If, for example, something binds at this place, say, no, I cannot eat food. It means that thing is competitive inhibitor. It competes with the substrate. But if, imagine if someone holds me from my hairs behind, I won't be able to eat food. That's the non-competitive. Non-competitive inhibitor binds at a place other than active site. Ali Abdullah, how will you manage? You'll have to watch very, very carefully yesterday's video.
So, why did we abandon the chapter?
Why did we?
Abandon the chapter.
What does it mean? Which one? Which one? No, biomolecule because people were not done with questions. और मैंने कहा मैं over the weekend उनको जो नूर और अरवा थी ना, I told them that I'll brief you this one, and the meanwhile we finished biomolecules. Yeah, we're done with it. वही ना, नहीं मैंने कहा जब उनको मैं ब्रीफ कर दूंगा आप लोग वीकेंड पे क्वेश्चंस कर रहे हैं फिर हम डिस्कस करें. सो फार जो हम कर रहे हैं वो हम ही क्वेश्चंस किए जा रहे हैं ना, बच्चे कर नहीं रहे. बायोमोलिक्यूल का तो लास्ट वाटर का टॉपिक कल किया ना हमने. नहीं कल पहले वाटर किया, जब जो भी लास्ट वाज वाटर, इट वाज ऑल डन. I don't know, but in this class I did water. Yeah, maybe day before yesterday we finished that. Now the idea is that I'll share with you. I have sent it. But the issue is that for every chapter I brief and then I do questions. That is not sensible. I want students do the questions. So chapter is not only one, but not only second, first is also not done well. Because proper way is that when the students do, when chapter is ending, they are doing questions, so we discuss them. Where are they finding hard? Because exam is also closer. No? Anyway, so right now we were doing inhibitors. So I hope the idea is done. You just joined me. You know, enzyme is a global protein. अब मैं आपको बताऊं ये एंजाइम से आगे हम तब चलेंगे जब स्टूडेंट्स के पास क्वेश्चंस हो. हम मंडे से वैसे भी बायोमोलिक्यूल्स के क्वेश्चन करेंगे और जिन बच्चों ने जो नए आए हैं दे विल वॉच वीडियोस ऑन व्हाट यू कॉल बायोमोलिक्यूल्स. जिन्होंने पढ़ लिया दे विल बी डूइंग क्वेश्चंस. सो मंडे डिस्कशन इज यूजफुल. A chapter doesn't take much time. Even in cells, it took us so much time. The reason was me teaching the idea, me doing the questions. तो वो सॉफ्ट कॉपी भी हम शेयर कर देते हैं. खैर एनीवे, बेस्ट थिंग इज सबा आप भी सांबा दें. बेस्ट सजेशन इज दैट यू शुड बाय द हार्ड कॉपी resources from the vendor Islamabad Photo State. I will give his address also in the group. So you should have that with you. So the classes are what we are doing in class, they should be with you as well. So I hope you understand what is competitive and non-competitive inhibition. Let me give a quick review that if imagine my head is all the enzyme, the globular protein, and mouth is what? Active site. So substrate is supposed to bind to active site. So if inhibitor binds, inhibitor is something that slows down enzyme action. This is what we did before you can. If inhibitor binds to active site, it means obviously substrate cannot bind. So this inhibition is called competitive or non-competitive? Competitive because there's a competition between inhibitor and substrate is of dash shape to the inhibitor. Yeah, same shape, similar shape. Complementary shape? No, complementary to active site is other. Substrate. Yeah, they have similar shape. So, but if you provide too much of, let me show you this, what we wrote before you came. So oh, I did not connect it. Maybe okay, let's have this diagram. Can you see? Okay, the idea is I hope it's visible now. Yes. Look, so if we increase the concentration of substrate, imagine this one is substrate, this guy is inhibitor. So if you increase the amount of substrate too much, then what would happen? That substrate will bind to active site, inhibitor won't bind. So competition will be one. So all the enzymes could bind to substrate only if we increase the amount of substrate too much. So this is called Vmax is achieved. Vmax is when all the enzymes got busy. नहीं समझ आ रही? Vmax means all the enzymes are engaged with substrate. So if you increase the amount of substrate too much, so probability, imagine in our class, if there are 99 boys and one girl, so there's possibility that boys' questions will be entertained because they are more in number. If there are 99 girls and one boy, girls' question will be entertained because they're more in number. So substrate is say so many molecules. Sorry? Probability. Yeah, probability, very good. So it's very much chance that substrate will bind and inhibitor won't bind. So this means this is what we say that Vmax is achieved. We can say that all these enzymes got busy with substrate. Please understand this very clearly for questions. Now, non-competitive binds to a place other than active site. No, this is substrate. This is non-competitive. Its shape is not as its shape is not similar to substrate. It will bind somewhere else. So unless this non-competitive inhibitor leaves, enzyme won't work, no matter how big is the substrate amount. So Vmax can never be achieved and non-competitive inhibitor. D-shaped, yeah. So now we go on to more explanation of this, what we did. There are two examples from your book. I would like to quote them. Very interesting. They say that, for example, you'll really love this example. You have to decide what kind of inhibition is it. So this is an example. I ask you to decide which example is it. What kind of inhibition? So imagine this is, what's this? Pen? Oh, so, very good. Very nice. So look, ethylene glycol, your book says it's a poison. It's something not good. Not poison, but it's not good. It will be converted to a poisonous substance. So its substrate, its enzyme that converts this has an active site. But if someone has drunk ethylene glycol, how doctors handle this? Doctors make him drink a lot of alcohol. Because the shape of alcohol shape of alcohol is similar to what is ethylene glycol. So what happens if just a chemical, I don't know. So if you add up alcohol would bind to the substrate, and the meanwhile ethylene glycol will be excreted out of the body. So my question to you is, what kind of inhibition is this? No, what is the inhibitor in this example? Okay, Sabha, आप क्या कहती हैं? Which one is inhibitor? Ethylene glycol or ethanol? Ethanol. So it is competitive inhibitor. It will bind to active site, and in the meanwhile, ethylene glycol will be gone out of body. Now next, there's another example that's very interesting. And this example is called end product inhibition. That again from your book. It's a very interesting example. It says that if there is an enzyme which works on the substrate, for example, starch, and which enzyme is it? Amylase. What? Amylase. Starch converts to maltose. Then there is an enzyme. You know which enzyme? Maltase. Works on maltose and makes what we call glucose. Let me show glucose by this. Yeah, you got it. So many glucose together make glycogen. Alpha 1416. Glucose is monomer, that's polymer. So look, so many glucose are made. What is it? आप टॉपिक को लॉक नहीं कर सकते जब तक आपके पास रिसोर्सेज नहीं है। चले एक दिन रखेंगे ना, विद ऑल क्वेश्चंस डन। भी देखो बहुत सारा ग्लूकोज बन गया है। एक्स्ट्रा की जरूरत भी नहीं है, रख भी लेंगे कि इफ नीड अराइजेस। नहीं कोर्स तो सही जा रहा है, क्वेश्चंस नहीं हो रहे ना बच्चों से। कोर्स तो डेफिनेटली सही जाना है, सही। द पॉइंट इज ये चैप्टर लॉक होगा जब क्वेश्चन होंगे, फिर हम डिस्कस करेंगे इसमें क्या रॉन्ग है क्यों। अच्छा बेटा देखो बहुत सारे ग्लूकोज बन चुके हैं, so body wants not glucose to be made so many, so some of the glucose molecules for example they go and want to, they go and bind to this enzyme. No, I should draw from upper side, excuse me. It's really hard to draw a line with this, but anyway, let me try if I could. So see, some of glucose molecules leave this place and go bind to what? Where should they bind? Active site or somewhere else? Somewhere else, because glucose does not match the shape of, this is amylase, na? Its substrate is starch. So glucose is not of same shape as starch. So glucose will bind to somewhere else other than active site. And this enzyme won't work, then maltose will not be made and no glucose will be made. So this thing happens when there is so much glucose and body wants to control the size of glucose. So what kind of inhibition is it? Because shape of glucose, सबा बताएं what kind of inhibition? Very good. Because shape of glucose is not similar to what? Starch. एक बात, दूसरी कंपटीशन नहीं है कि आप जितना मर्जी स्टार्च कर दें, जब तक ग्लूकोज को छोड़ेगा नहीं, एंजाइम वोंट कन्वर्ट स्टार्च टू maltose. So there's no competition. It's not that if you increase the amount of starch, glucose, enzyme will make glucose. No, enzyme will work, amylase will work only if glucose leaves it. So there's no competition. Enzyme will work only if glucose leaves it. So Vmax can be achieved or cannot be achieved. G saba V velocity rate. G saba Vmax can be achieved or cannot be achieved. Very good. So this is called non-competitive inhibition, not similar to substrate. Vmax can't be achieved. And this is called end product inhibition. That when end product inhibits the reaction, enzyme. End product was glucose, that is inhibiting the enzyme. So end product inhibition is the example of what? Competitive or non-competitive inhibition? Non-competitive. Now I'll be carrying you to some questions.